Today I'm going to discuss the steps that occur during muscle contraction. Muscle contraction occurs due to the sliding filament theory. This theory suggests that the myofilaments, which are the fibrous strands that carry out the contraction process, do not actually become shorter. Instead, the thin filaments slide over the thick ones, pulling the Z discs behind them. The Z discs are what provides the anchorage for the thin filaments. This sliding process causes each individual sarcomere to shorten as a whole. The sarcomere is the functional contractile unit of the muscle fiber. The contraction process begins with the arrival of the nerve signal. The signal arrives at the synaptic knob and opens the calcium channels, allowing calcium ions to enter. The presence of the calcium causes the synaptic vesicles to release the acetylcholine, or ACH, into the synaptic cleft. The acetylcholine diffuses across the cleft and binds to the receptors. This opens ion channels that allow sodium ions to flow into the cell and allow potassium to flow out. The ion flow causes the polarity to reverse and create an action potential. The action potentials spread down from the motor end plate to the T-tubules, then continue down the T-tubule to the cell interior. In the T-tubules, the action potentials open voltage-gated ion channels that are linked to the calcium channels in the sarcoplasmic reticulum. The calcium diffuses out of the sarcoplasmic reticulum, down its concentration gradient, and into the cytosol. The calcium then binds to the troponin of the thin filaments. When the calcium binds to the troponin tropomyosin complex changes shape and exposes the active sites on the actin filaments. The exposure allows the filaments to bind to the myosin head. Energy released by the hydrolyzation of ATP that is bound to the myosin head activates the head and causes it to become extended in a cocked position. The ADP and phosphate that was released by the ATP stay bound to the myosin head at this point. Next, the cocked myosin binds to an exposed active site on the filament. This binding forms a cross bridge between the actin and the myosin. After the cross bridge is formed, the myosin releases the ADP and phosphate and flexes. When the myosin flexes, it moves to a bent position and brings the filament with it. This motion is called the power stroke. Finally, ATP will bind to the myosin head again, destabilizing the myosin-actin bond. The ATP binding breaks the cross bridge and the process is ready to be repeated.